Okay, if everyone could please be seated. Welcome everybody to the Burnet Oration. The Burnet Oration is one of the major events in the ASI calendar. It provides a forum for one of our outstanding scientists to give us a talk about not only their research but their memories of their various steps in their career and their career highlights. Often the Burnet Oration includes some personal content where the speaker might reflect on the context of the research at the time when they were doing it, the people and the events that influenced them as they progressed up the career scale. And where possible, they're encouraged to relate their experience to the great scientist Sir Frank Macfarlane Burnet. It's getting increasingly difficult to find people who worked with Burnet, let alone had met him. But regardless, the Burnet Oration is a wonderful annual event to honour the memory of Burnet and all of the things that he contributed to immunology and to our society. We're also very grateful to CSL Limited for being our long-term sponsor of this event. Now, inviting and introducing the Burnet Orator is one of the great honours of being the president of ASI. And I actually admit I felt quite nervous about this, and so I spoke to our past president, David Tarlington, who's always a source of wisdom for me with these things. And he looked me straight in the eye and he said, "Dale, this is a very important event. You're a guest in New Zealand here, so don't get into this Australia-New Zealand sledging that Graham sort of introduced this morning. You know, for example, don't mention the America's Cup." And I said, but that was an American boat that beat the New Zealanders at the finishing line. And David said, yes, but it was an Australian skipper. And that's probably going to upset them. So I said, oh, fair point. But it'd be worse if I mentioned the Rugby League World Cup victory yesterday. And he said, oh, I didn't see that. What happened? I said, oh, Australia, the Kangaroos beat the Kiwis 34-2. And he said, oh, that's a terrible loss for New Zealand. I said, yeah, but it's a terrific loss for Australia. He said, yeah, well, definitely don't mention that either. So, am I going all right so far, David? Anyway, John Fraser, I'll move on to the subject of this meeting. John Fraser has had a career of over 25 years in the field of immunology, and he's worked in other areas of science prior to that. But while he's been in immunology, he's been a long-standing member of the Australasian Society for Immunology and also a key contributor at various events that we've held. Some of the highlights in John's career: in 1992, he was the inaugural New Zealand recipient of the Wellington Trust Senior Research Fellowship. In 1998, he was appointed as a professor in the molecular medicine faculty of the University of Auckland. Professor in molecular medicine at the University of Auckland. And in 2005, he was appointed a Royal Fellow of the Royal Society of New Zealand. And he's currently the Dean of the Faculty of Medicine and Health Sciences. And last year, in 2012, he was given the honour of the Sir Charles Hercus Medal from the Royal Society of New Zealand for his contributions to immunology research. A major focus of John's research has been the mechanisms of microbial pathogenesis and the interaction of the immune system with two major and very dangerous pathogens, Staphylococcus aureus and Streptococcus pyogenes. John's had many outstanding achievements during his career and many outstanding contributions, and one of the highlights was the seminal discovery of the molecular basis and the molecular mechanisms by which bacterial superantigens cause toxic shock syndrome. John's widely regarded as the international expert on superantigen research. I won't go into any more detail about John's achievements because I want to leave something for him to talk about. So it really just remains for everybody to please join me in welcoming the 2013 Burnet Orator, Professor John Fraser, to the stage. Thank you.
Thank you very much, Dale. And thank you very much for the Council of ASI for considering me worthy of this singular honour of the ASI. Of course, I've been with the ASI for many, many years, and I have sat through many Burnet orations, all of which I have thoroughly enjoyed. And it comes as some amazement that I'm standing here today as the 2013 Burnet Orator. So I hope I can live up to past histories. I would first of all just like to again thank Commonwealth Serum Laboratories, who I know has supported this very important lecture for the ASI for many years. So what I thought I would do is as most Burnet orators do, reflect a little bit on career and influences, and then perhaps talk a little bit about the science that I'm most interested in. And I realise that immunology covers a wide area, and it's a relatively rarefied and unique area that I've worked in, but it's one that I've loved, and I've become quite passionate about some of the ways in which we could take the information, the basic science, and then turn that into translational achievements, because of course we're all here to achieve advances in the health of human beings. So my title is A Reflection of 40 Years of Immunology Down Under, and it's a rather grandiose title because it's actually not 40 years of immunology, it's 40 years in science. My immunology is about 30 years, and I have to say this year I was in Boston meeting with my old postdoc supervisor, who I will talk about a little bit later, and he invited me to sit in on one of his seminars with his undergraduate students at Harvard, and he introduced the grey-haired gentleman in the corner as a fellow that used to be in his lab 30 years ago. And it was one of those holy shit moments, which made me realise that 30 years had passed, and I was quite taken back by that. But of course, Jack had been in science for 60 years, so I was only a child in comparison to his contribution. So Macfarlane Burnet, we all know about Macfarlane Burnet. I never had the opportunity to meet him. I certainly met with some of the people who work with Mac Burnet, in particular Gus Nossel. And I won't go into detail because everybody knows that Macfarlane Burnet was probably one of Australia's greatest scientists. He really singularly was one of the fathers of modern immunology, along with Peter Medawar. He was a clinician, obviously, and was well known for winning the Nobel Prize with Medawar in 1960 for the theory of acquired immune tolerance. Of course, he's best known for his development of the clonal selection theory, but he also worked in other areas, so he was clearly a dedicated medical scientist. He was interested in disease, and he was interested in resolving disease, and clearly his interest was in diseases that were associated with the immune response or the failing of the immune response. But he did a lot of other things. Particularly, he developed the hemagglutination assay for culturing and detecting influenza, which was no mean contribution to medical science. And also developed methods for growing influenza in hen eggs, which is, of course, the way in which we grow influenza for vaccines today. He was an absolutely prolific writer, and I believe he dedicated 1 day a week to writing, a lesson for you PhDs and postdocs. The dedication to writing, I think, serves all scientists well. He was an avid public campaigner, one of the first public campaigners against smoking and nuclear proliferation. And not to give you the impression that he was all still a light, he was also known for his strident attacks on the usefulness of molecular biology and cell biology. I think we all agree that nobody's perfect. One of the things that intrigued me most about Macfarlane Burnet was this remarkable honest admission by him, which was really to admit that, in fact, his contribution to one of the more important discoveries in immunology in the 20th century was, in fact, a very minor one. And minor in the fact that he really only provided the hypothesis that called for an experiment. Now, these days, one would argue that a hypothesis doesn't get you anywhere, but talking to Phil just earlier, we were waxing lyrical about how science is devoid of hypotheses these days. And in fact, you could win a Nobel Prize simply on dreaming up and thinking up a really good idea and then leaving it to other people to test. So, of course, I looked at the website with all the Burnet orators, and they are a quite remarkable group of individuals, and I feel most privileged to be considered amongst these. And of course, most of these will be known to you. One of the things I was struck by this, however, is that, and I'm not sure you realise or notice, that they do seem to be getting a little younger as we move on. And that's a very warm reference to you, Phil, of course, as the last Burnet orator. So I thought I would seize the opportunity to add my photo. just to show you that I'm actually nearly used to have grey hair. In fact, I had quite curly hair, dark curly hair. However, so my roots, as you probably detected this morning with my Maori and singing, I am actually part Maori and very proud of that fact. And so I thought I would show you this wonderful photo from 1898. This fellow here is my great-grandfather by the name of Horiwaka, or George Walker, because the English didn't know how to say Maori back then. And he married this very dour Irish woman, Margaret, and they had many sons and daughters. In fact, they had 12, and that's my grandmother. You can't really see on the photo, sitting on the knee. She was born in 1898. And Hori had a brother, George, who had 5 wives and 16 children. He was very busy. And they all survived, and wonderful large family, extended family. Hori is in fact the son of probably the first mixed marriage in the Waikato region between my great-great-grandfather who came out from Scotland, and he married a Maori woman, Raurangi Takari. And so they were one of the first mixed marriages in all of New Zealand, in fact. This is my father here, who's descended from James Walker, and my mother, Hope. She's still alive. My father was a businessman, and during my days when I was a PhD student and then a postdoc, he would regularly remind me and ask me when I was going to get a real job. I think I disappointed him entirely. I think he finally gave up on that idea when I got an appointment at Auckland and he realised that his son was going to be an academic, which was only slightly more prestigious than failing musician, which was my other career plan. This is taken from my Hutt Valley High School days, and of course Hutt Valley High School is just a little way out in Wellington, in Lower Hutt here. It's actually a very old school, and my grandfather actually built, or was one of the engineers on building this. Well known in the science fields, of course, because Alan MacDiarmid, who was one of our few Nobel Prize winners, went to school, actually in the class with my father, class of 44. This is me back in 1975, and you can see we didn't really have any hair rules in those days. Extraordinary length hair. It was all the rage to wear our hair that long. Thank God that's gone away. And that's me in the corner here, dreaming about things unscientific, I have to admit. I wasn't really that interested in science, although I did reasonably well at school in science. It wasn't a career that I thought I would end up doing, but I went to university anyway. And I suddenly realized in doing this lecture that I have been a permanent academic for 40 years. And I'm not sure that's a proud admission or one that I should be slightly bemused by, but I did my undergraduate degree at Victoria here, then a PhD at University of Auckland, and I freely admit that having done my PhD, I was very nonplussed by the idea of science. I'd done a PhD mainly because I didn't really know what else I should do. And of course, I was really unsure as to what direction I would take, until the head of the department, Alistair Rennett, came to me and he said, well, would you like to go and work with Jack Strominger? And I said, Jack who? And he said, well, I had another student of mine at Cambridge named Peter Parham, who I sent to Jack Strominger, and he did very well. So I'm sure if I picked up the phone and talked to Jack, he would listen to me. And I said, sure, why not? Because I was between jobs at that time. And he certainly did so, and I got a call from Jack asking if I ever wanted to come and do a postdoc. And in those days, it was relatively easy to get fellowships to go to the United States. Something that I think we probably lack today is the ability for young people to move seamlessly from PhD to postdoc, which I think, in my mind, is probably the most important transition you can make. So I packed my bags in 1983 and went off to Harvard University to work with the great Jack Strominger. And it was really my probably my most important career decision of my life. I didn't know much about immunology when I was a student. I did read this paper when I was doing a PhD. It was by Alice Reinherz and Stuart Schlossman, and it was a transformative paper for me because it was a review of the state of art of immunology at that stage, and it just caught my attention. It caught my attention for five things, and I can still remember what those 5 things were. First of all, it described the ability that you could actually clone T cells. You could make individual cells that had unique phenotypes using new growth factors that had been isolated. Not only that, you could immunize mice with these clonal cells and then take their spleens out and make monoclonal antibodies, and these monoclonal antibodies would recognize individual molecules on the surface of these cells. And this was a great way of separating and distinguishing these cells from each other and finding out what their functions were. And of course, the newly invented technique called fluorescent activated cell sorting made this even easier. And the upshot of this paper was that they had discovered that T cells were actually divided into two populations that they called T4 and T8. T4 was what they referred to as the helper, which produced cytokines, and T8 was known as the cytotoxic suppressor phenotype. So this was quite interesting to me. I thought, well, this is technology that I think is new and interesting, and certainly something that I could get involved in. So I was awarded a Fogarty Fellowship and went to work with this great man here, Jack Strominger, who remains a very dear friend to me today. And those of you who don't know Jack Strominger, he is really the godfather of MHC. He is renowned for his work initially in the area of bacterial cell wall synthesis, almost single-handedly working out the mechanism of penicillin in the inhibition of the pentaglycyl bridge that forms the cell wall of gram-positive organisms. His first paper was published in 1953, so he's been publishing since for 60 years. And in fact, he has over 1,100 publications in PubMed now, some of them some of the most important publications in immunology. Of course, he's best known for his genetics and biochemistry of MHC, and I was fortunate enough to arrive in his lab when this was the main theme. And of course, with Pam Bjorkman and Don Wiley, resolved the structure of MHC HLA-A2 and then later the DR1 in the late 1980s, which was one of the most important structures to be resolved, I believe, in all of science. And then he went on and has worked and had a long career in studying NK receptors and innate immunity in pregnancy. Quite importantly, and something that he's most proud of, he's trained over 300 postdocs and PhD students. One of them is now a Nobel Prize winner, and I think 11 of them are members or fellows of the National Academy of Sciences. So he's probably more proud of that achievement than any of his scientific achievements. And of course, he's won many different awards. But the thing that he says was most important in his strategy in running a lab was that he was permissive. He allowed all sorts of things to happen. And certainly my experience there was a wonderful experience. You could take an idea and you could develop it, and you wouldn't get the, what are you doing that for? I'm not spending money on that. He was extremely permissive. It was a very large lab, I admit. He actually had 2 labs. I think in total, when I was there, he must have had about 20, 25 postdocs, all working on different areas. But it was hugely productive and very exciting. So I just thought I'd list some of the memorable Jackisms. There are lots of stories. In fact, those who have worked with him, one day we thought of actually writing a book of all the stories about Jack. We never got round to it, but here are some of the things that I remember the most that he used to tell me on several occasions. First of all, you'll never make it in science unless you publish, and I think that's probably pretty obvious, but a lot of us forget that the importance of writing is just as important as doing the experiments. Another one he used to tell me, there's no such thing as good journals, only good papers, which is absolutely true. But then he'd turn around and say, don't publish there, it'll get buried. The one that I remember the most, and the one I tell my students, is to be successful in science, you have to focus. And I think that's as more important today as it is back then, where you have to pick a topic and work on it and thoroughly immerse yourself in it and be good at it and be known about it. And he's absolutely right. Another one that I remember distinctly is, if it's important, someone else will have found it too. And I have experienced that on a number of occasions, and I'll give you a good example. And he would always ask us, whenever we were doing an experiment and we were showing the data, he would always ask, what's the big question here? I think the importance of remembering why you're doing science and what big question you're asking is more important, in fact, than actually doing the experiment. So this is just a picture of Jack recently with his lovely wife, Anne. This is in the faculty club at Harvard. Jack is now 87 and has every intention of being the oldest living scientist in America. He's determined to make it to 90. And as I said, I was privileged to sit in on a seminar that he gave to students about 3 months ago. And after the seminar, he looked at me and said, I think that's the last seminar I'm going to give. And I said, why, Jack? And he said, I couldn't hear what they were saying. However, so I've spent about 30 years from I started my postdoc today in immunology. It's an area that I have thoroughly enjoyed. I've watched a lot of things happen. I have been involved in some of them, and I've tried to sort of break them down into 3 epochs. Certainly my memories of the 1980s was clearly, it was the age of molecular immunology. It was the era when molecular biology suddenly hit immunology, and all of the theoretical stuff was thrown out of the window when people could now clone the genes that we only dreamed about. And certainly my memories were that it was a wonderful time in immunology where structures and genes and functions all became apparent in very, very rapid succession. So it was all about adaptive immunity, and I have to say, if you worked on things like innate immunity and complement back in the 80s, editors were not really interested. That changed dramatically in the 1990s, of course. That was the decade of tolerance memory and, of course, pattern recognition. And I want to just talk briefly about that, my experience of the development of those ideas, particularly with Charlie Janeway. And then in 2000 is clearly the decade of immune regulation and the development of, or our better understanding of inflammation. So that's my reflection on 30 years of immunology. I'm sure you might have other reflections, and I'm sure there are areas that are missed out. Those are the things that I remember the most. The one thing I do take note of, or would like you to take note of, is this little joke that I tell people whenever they, and I'm not sure immunologists actually see the humor in this, but I think it's true. And it's about reductionists and reductionist science. And I freely admit to being a strong advocate of reductionist science myself. But in immunology, of course, it's really important to remember the bigger picture and the issue that you're working with a very, very complex interreactive system. So this is a joke about frogs where the immunologist is trying to identify how frogs hear, and he pulls off the legs, of course, and he comes to the wonderful conclusion that the ears are located in the legs. And of course, that's always the risk in immunology, is if you take things apart and only work on one section, you're actually missing other aspects of what's going on. And this is no better true in my field, of course. And of course, there are a lot of people in this audience that will probably take some slight at this comment, but of course, mice aren't humans. And there are many avenues and areas where I can identify where things work in mice, they don't work in humans. And it's been a problem for me, of course, because the area that I work in, particularly in the area of staph and strep, it's most important because staph and strep are not organisms that have evolved with mice. In fact, they've evolved in primates. So my favourite organism is Staph aureus. It's probably one of the most important pathogens on the globe today. It's certainly the pathogen that creates potentially the greatest risk of becoming the superbug that we're all in fear of. The important thing about staph that you have to remember is, in fact, it evolved primarily in primates. So most of the toxins and virulence factors that these organisms, this microbe produces, that interacts with the immune system are highly specific and only work in humans, which makes studying them extremely difficult because you can't breed mice to do your experiments for you. And it's very difficult to breed humans to do the types of experiments you want to do. Ethics committees have slightly different views on those types of experiments. So that has been a significant problem in our area, and it's still a problem we face today, is that the things we want to understand about these organisms and the way they interact with, particularly the innate immune response, become problematic when we can't use model systems like mice or rats. So I'd just like to tell you about some of my aha moments. These are areas that I can remember distinctly that changed my way of thinking and really influenced the way in which I worked. So the first one, of course, I've already talked about was that wonderful reflection by Alice Reinhertz and Stuart Schlossman around what was known at the turn of the 1980s around the differentiation and function of T lymphocytes. Probably the most important thing that happened in my scientific career was the next one, which was the introduction of the Maniatis Bible, molecular cloning. And of course, that changed a lot of people's lives, and I think that's actually cited as one of the epochal moments in science in general, because it allowed all of the technologies and methodologies that could only have been done in the Cold Spring Harbor laboratories to be done in all sorts of laboratories around the world, including ours. And it actually turned out to be really simple and really easy to clone genes and to do all of the genetic experiments that were available at that time. And so we got deeply involved in using molecular biology to understand the mechanisms of the immune system. Of course, I arrived in 1983, and the T-cell receptor genes and proteins were discovered by a number of groups in 1983 and 1984. Alice, of course, John and Pippa, Jim Allison all had monoclonal antibodies to the T-cell receptor, idiotypic monoclonal antibodies, and of course Mark Davis and TAC-MAC, and then Susumu Tonegawa cloned the genes. So it was all on. So T-cell immunology went from complete non-understanding of how T-cells work to suddenly having the receptor that we could study. Of course, the structure of MHC was another aha moment, because when we first saw that, we suddenly realized what was actually going on. And of course, I did want to mention Charlie Janeway, who I had a lot to do with at the end of the 80s and early 90s, because he was also very interested in superantigens and MLS, myelolymphocyte stimulating antigens, which he very cleverly decided were behaving in the same way. And he actually wrote a very nice paper before anybody else had worked on them, describing his theories on how MLS and superantigens were working in the same fashion. But of course, his most famous paper was the paper which outlined his ideas around innate immunity and the need for having pattern recognition and pattern recognition receptors. And it's one I remember talking to him at length over in a number of discussions, and it was the most wonderful, evocative. Those of you who remember Charlie or knew Charlie will realize what an extraordinary mind he had, and he always seemed to be about 2 or 3 steps ahead of everybody else. Very sad that he passed away. However, my most memorable aha moment was this one here. I would like to say that I met Tam, my beautiful wife, while walking through Central Park one day. Found this girl sitting on the lawn. However, it occurred slightly less memorably than that. We met in a shop, and I remember taking her out for dinner one night to convince her that I was worth dating. And I remember describing to her what I did, and using a plate of noodles to describe molecular cloning. I believe it worked. I think she understood what happened, but she seemed to get so excited that the mushroom that was on the end of her fork flew across the room and hit some poor guy in the corner. So it was a memorable evening, and obviously I made enough of an impression on her for her to allow me to drag her back to New Zealand, where we were married. And we have produced, I admit in the right order, 2 absolutely beautiful girls, Ashley and Rachel. Ashley on the left and